Hello and welcome to this introduction to microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the ways in which we can fix and stain cells for light microscopy. And a key question is, why might we want to fix and stain cells? Well, in most cases, increasing contrast is essential for improving the detection of cells via light microscopy, as many biological specimens are nearly transparent under normal light. Enhancing contrast not only makes structures more visible, but also helps to increase resolution, allowing finer details to be distinguished. And so to achieve this, we use two key techniques: fixation to preserve cell structures and staining to selectively highlight different components. These methods enhance the visibility of microorganisms and cellular structures, making them easier to study under the microscope. Now, one of the first stages in microscopy is fixation, where a biological sample is immobilized on a microscope slide. This prevents it from moving, allowing us to focus clearly and capture a stable image. And as part of this process, the sample is effectively killed, meaning all biological activity is halted. This essentially preserves the sample as close to its natural state as possible, preventing decay or distortion. However, some degree of cellular damage may still occur. Now, there are two common methods used to fix biological samples for visualization under a light microscope. We have heat fixation and chemical fixation, each with its own advantages and limitations. And so when samples are heat fixed, the sample is air dried before being passed through a flame or placed on a hot plate. This method is simple and quick, but it can cause cell distortion or shrinkage, making it less suitable for delicate structures like capsules and flagella. On the flip side, we have chemical fixation. This is where the samples are also air dried on a slide, but instead of heat, a fixative chemical such as methanol or formaldehyde is applied. This process takes more time and is slightly more complex, but it reduces the overall distortion, preserving finer cellular details. Now, each of these methods has its pros and cons depending on the structures we want to visualize. However, for most routine microbiology, we heat fix samples. And how do we do this? Well, first we take a glass microscope slide and place a small drop of liquid culture onto the center using a sterile loop or pipette. The cell culture is then spread into an even layer and allowed to air dry completely, which typically takes a couple of minutes. Now, once it's dry, high heat is directly applied to the sample in order to fix it. Now, as I said, there are two different methods. The most common uses a Bunsen burner and passing the glass slide through the flame 2 or 3 times. The alternative method is to place the glass slide face up on a 60°C hot plate for around 1 to 2 minutes. Now, this heating process kills the cells, adheres them to the slide, and preserves their structure for staining. However, it also minimizes sample loss during our next preparatory stage, which is staining. And this involves applying a dye to our sample which enhances contrast and improves the visibility of cellular structures. Now, since most cells are naturally transparent, it can be difficult to distinguish details under a microscope. However, stains bind to specific cellular components, allowing us to visualize different aspects of the cell with greater clarity. Now, there are two different types of stains we can use. Simple stains use a single dye to uniformly colour all cells, increasing contrast to make them more visible. And a common example is methylene blue, which highlights the overall shape and structure of cells. Differential stains, on the other hand, use multiple dyes applied sequentially with wash stages in between, and these stains typically allow us to distinguish different cell types, providing more detailed information about a sample while still increasing contrast. The Gram stain is a classic example of this and is used extensively in bacteriology to differentiate bacteria based on their cell wall composition. Okay, so now we'll go through both staining techniques in more detail, looking at how samples are prepared and how they appear under bright field microscopy. starting with methylene blue, which is a simple stain used to enhance contrast. This stain works by binding to negatively charged components within the cell, such as nucleic acids and cell membranes due to electrostatic attraction. And so by staining the entire cell uniformly, methylene blue makes it easier to observe cell shape, size, and arrangement, helping us to identify key structural features under the microscope. So let's have a look at this process. And so to stain a cell sample with methylene blue, we start with a heat-fixed specimen on a microscope slide. Next, we cover the sample with 1% volume per volume methylene blue and let it sit for around 60 seconds. This allows enough time for the dye to penetrate into the cells and bind to those negatively charged components, such as nucleic acids and the cell membrane, ensuring efficient staining. And so once the cells have been stained, the next step is to rinse off the excess methylene blue using water. This removes any unbound stain while keeping the cellular structures clearly visible. And after rinsing, we then allow the slide to air dry, and if needed, we can gently blot the surface with absorbent blotting paper to speed up the drying process, being careful not to smudge the sample. And so now that the sample is prepared, there is only one thing left to do. We apply a glass cover slip and view it using bright field microscopy on a compound light microscope. And so in this example, at 1000 times total magnification, we can see Streptococcus pneumoniae cells within a cerebrospinal fluid sample. And due to the methylene blue, the bacterial cells appear blue against a translucent background, clearly highlighting their shape and arrangement. Now unfortunately, this spells bad news for the patient, as cerebrospinal fluid should be sterile, and so its presence here indicates a serious infection such as bacterial meningitis. Now our next stain is a little more involved because it is a differential stain, and in this case we're looking at the Gram stain, one of the most widely used techniques in bacteriology. The Gram stain, originally developed by Hans Christian Gram in 1884 and always written with a capital G, allows us to distinguish bacteria into two major groups based on differences in their cell wall composition, referred to as either being Gram-positive or Gram-negative. Now this classification is crucial for bacterial identification and antibiotic selection. Now because it's a differential stain, we use two different dyes applied in a specific sequence to highlight these structural differences, and these are crystal violet and safranin, which work together to differentiate Gram-positive from Gram-negative bacteria based on their cell wall structure. Now as a slight recap, the Gram stain highlights differences in cell wall structure between Gram-positive and Gram-negative bacteria. Gram-positive bacteria have this thick peptidoglycan layer and only one cell membrane. Now cells with a thick peptidoglycan layer are able to retain crystal violet within this structure, staining them purple and earning them the name Gram-positive. Gram-negative bacteria, on the other hand, have a double membrane structure with a thin layer of peptidoglycan. Now cells with this thin layer are unable to retain crystal violet, and so they will take up the second stain safranin, appearing pink or red under the microscope and earning them the name Gram-negative. Okay, so to stain the cell sample with the Gram stain, we start with a heat-fixed specimen on a microscope slide. Next, we cover the sample with 0.5% volume per volume crystal violet solution and let it sit for around 60 seconds. This allows enough time for the dye to penetrate into the peptidoglycan layer in our bacterial cells, ensuring effective staining. Now once all the cells have been stained, the next step is to fix the crystal violet in place using iodine, and so we apply this to the sample and let it sit for another 60 seconds. Now during this time, iodine acts as a mordant, reacting with crystal violet to form a crystal violet iodine complex, which becomes trapped within the peptidoglycan layer. Now this process is effective in Gram-positive bacteria because there is a thick peptidoglycan layer reinforcing that purple stain. However, in Gram-negative bacteria, the thin peptidoglycan layer is unable to retain the complex as strongly, making them susceptible to decolorization. And this just happens to be our next step. We apply 95% volume per volume ethanol to the sample. And this step is critical. It removes the crystal violet iodine complex from our gram-negative bacteria while leaving gram-positive bacteria stained purple. Now the timing for this step is crucial. Over-decolorizing can strip crystal violet from gram-positive cells, making them appear gram-negative, whereas under-decolorizing may leave gram-negative bacteria still retaining some crystal violet, leading to false results. Now before we can continue, the sample needs to be washed with water. This is another crucial step as it removes any remaining ethanol, effectively stopping that decolorization process. And so without this wash step, ethanol could continue stripping that crystal violet stain from the sample, potentially leading to false negative results in our gram-positive bacteria. Now once the sample is rinsed, the slide is ready for the final staining step. This is where we counterstain the sample with safranin. Now just like the previous steps, we apply our 0.5% safranin to the sample and let it sit for around 60 seconds to allow the dye to penetrate into the peptidoglycan. At this stage, gram-negative bacteria, which lost the crystal violet stain, take up the safranin and appear pink or red. Gram-positive bacteria can also take up safranin, however they remain purple as the intense crystal violet stain masks the pink counterstain of safranin. And so once the cells have been stained, the next step is to rinse off the excess safranin using water, removing any of that unbound stain. And so after rinsing, we then allow the slide to air dry, and if needed, we can gently blot the surface with absorbent blotting paper to speed up the drying process, being careful not to smudge the sample. Now the sample is prepared, there is only one thing left to do. We apply a glass cover slip and view it using bright field microscopy on a compound light microscope. And so in this example, at 1000 times total magnification, we can see a mixed bacterial population, with some gram-positive bacilli staining purple and some gram-negative bacilli staining pink. And this provides us a very simple and quick way to differentiate between the two main types of cell walls in bacterial cells. However, a minority of bacteria are resistant to the gram stain, and this is due to the presence of a waxy lipid-rich cell wall that contains mycolic acid. This essentially prevents the penetration of water-based dyes such as crystal violet, and so in this instance we would opt for a different differential stain such as the acid-fast stain. Now I won't go into the full process of this stain, however it involves the use of carbonyl fusion, which penetrates the lipid-rich cell as well as other cells, a decolorization stage with acid which removes carbonyl fusion from non-waxy cells but not from acid-fast bacteria, and brilliant green as a counterstain, allowing non-acid-fast cells to be distinguished while acid-fast bacteria retain that red colour. We would then visualize these samples using bright field microscopy on a compound light microscope. And so in this example, at 1000 times total magnification, we can see Mycobacterium cells appearing purple because they retain that carbonyl fusion dye, even after the acid decolorization step, which is due to their waxy, mycolic acid-rich cell wall, which prevents the stain from being washed away. In contrast, the Staphylococcus cells appear green, as they do not retain the carbonyl fusion and instead take up the brilliant green counterstain. And so this particular staining method is crucial for identifying acid-fast bacteria, such as those responsible for tuberculosis and leprosy. Now some bacteria, such as various Bacillus and Clostridium species, can produce highly resistant spores, allowing them to survive harsh conditions. These spores have a tough outer layer that makes them very difficult to stain with conventional methods. And so to visualize spores, we use the spore stain technique. This is where malachite green is applied and driven into the spores using heat, allowing the stain to penetrate the tough spore coat. The sample is then decolorized with water, which removes malachite green from non-spore structures while the spore remains stained. And finally, a safranin counterstain is then applied, staining the vegetative cells pink while the spores remain green. Now, this technique helps distinguish spore-forming bacteria from non-spore formers, which is important in both clinical and environmental microbiology. We can then view these cells under bright field microscopy on a compound light microscope. And so in this example here, at 1000 times total magnification, we can see Clostridium difficile, a well-known spore-forming bacterium. Here, the vegetative cells appear red as they have taken up the safranin counterstain, and in contrast to this, the endospores remain green as they retained that malachite green stain. Now, this clear distinction between spores and vegetative cells highlights the power of the spore stain in identifying spore-forming pathogens, which are often resistant to many growth control mechanisms such as heat, chemicals, and antibiotics. And our final staining method is the negative stain, a technique that stains the background rather than the cells themselves. Now, this method uses an acidic dye such as India ink, which is repelled by the negatively charged bacterial cell surface, and as a result, the background appears dark while the cells remain unstained, creating enhanced contrast without penetrating the cell. Now, a common question is, why would we want to use a negative stain? Well, it's particularly useful for visualizing structures that do not readily take up traditional stains, most notably the capsules of bacteria, which can play a key role in bacterial virulence. And so if we were to use bright field microscopy on a compound light microscope, we could then visualize these cells. And so in this example, at 1000 times total magnification, we can see Acinetobacter chalcoaesticus, an opportunistic pathogen known for causing nosocomial infections such as pneumonia, wound infections, bacteremia, and even urinary tract infections. Now, in this image, we notice a clear zone surrounding the cells where the dye has been excluded. This highlights the capsule, a polysaccharide or protein layer that provides protection to the cell and aids in immune evasion. It can also enhance adhesion to surfaces within a host. All of these are key factors in its ability to establish an infection. And so, as you can see, negative staining is one of the best ways to visualize bacterial capsules, which play a critical role in pathogenesis and antibiotic resistance. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.